So moving on to the reproductive system. So today we'll cover things that they have in common. And then next week we'll cover male reproductive system and then female reproductive system. Male and female reproductive organs share the same 4 tasks. The one that we're going to go over a little bit today is forming gametes. So we'll go over meiosis. And gametes are specialized cells for sexual reproduction. They're called sperm in males and ova or eggs in females. So why are they special? They're special for 2 reasons. One has to kind of to do with number 3 a little bit, but the 2 specializations of gametes is the first one is that they are haploid. And the second is that they have an increase in genetic variability. So in our bodies, we have 23 pair of chromosome. These are called homologous chromosomes, meaning that our diploid number is 46. So we have a total of 46 chromosomes. We have 23 pair. For, let's just say chromosome number 2, okay? Our cells, like our body cells, our skin cells, our heart cells, our kidney cells, our liver cells, our brain cells, any cell with a nucleus is going to have the same DNA. So that's why you can take any cell you want, sequence it, and you'll get somebody's DNA. For those 23 pair, we got 1 chromatid from mom, and we got 1 from dad. Okay, so if we're looking at hair color, so my mom is blonde, so I probably got like a blonde. They're called alleles. So an allele, we have 25,000 genes. We have 23 pair of chromosomes. So you do the math, and some chromosomes are larger, some are smaller, some genes are larger, some genes are smaller. But for simplicity's sake, we'll say there's like 1,000 genes per chromosome. So if we're looking at chromosome 2 or chromosome 8, I don't know where hair color is on, but a little part of that chromosome is going to code for hair color. And so I got 1 from my mom and I got 1 from my dad. My dad has black hair, my mom has blonde hair, and you can see I have brown hair. So the wonders of genetics. When we make gametes, we need to get down to a single set of 23 because we need the zygote, which will turn into the embryo and the fetus and the baby, to have the same amount of DNA as a normal human does. So our gametes are going to need to be haploid, where they have a chromosome number of 23. So the sperm will have 23, the egg will have 23, and when they form a zygote, we'll get 46. But it's not simply dividing and not simply replicating and then dividing, because then all of that chromosome would come from either, like for my kids, would either come from like my dad or my mom. And what we want to do is we want to increase genetic variability. So there's going to be something called crossing over, where different chromatids switch genes. And so no chromatid is going to be solely maternal or solely paternal for my contribution. And I've got pictures to show all this, but it's increasing genetic variability is going to be a key, one of the most important things about sexual reproduction, because it gives population variability and a greater survivability. Okay. Back to the 4 tasks. So forming gametes, specialized cells for sexual reproduction, bringing gametes together through sexual intercourse, combining genetic information through fertilization, and then supporting development of a fetus and birth of the baby. If you were to, how you like go on those shows and they're like, oh, are you smarter than a 5th grader? If you ask most people what their primary sex organs are, they'll go to external genitalia or internal genitalia. But in reality, if you're an A&P'er, you know that the primary sex organs are the testes in male and the ovaries in females, because they produce the gametes and sex hormones. Okay, so gametes are sperm in male, ova in females. Gametes are formed by cell division called meiosis, which we're going to go over. They also produce sex hormones. So the testes produce testosterone, the ovaries produce estrogen and progesterone. These sex hormones are vital in development and function of reproductive organs and other organs or tissues, sexual behavior, and sexual drive. Accessory reproductive organs are ducts, glands, and external genitalia. Okay, back to the hypothalamus. So production of gametes and sex hormones is regulated by the hypothalamic anterior pituitary gonadal axis, meaning that the hypothalamus is at the apex. It is going to secrete gonadal releasing hormone, so a stimulatory signal, via the portal system so that the concentrations stay high to the anterior pituitary. The anterior pituitary secretes luteinizing hormone and follicle stimulating hormone, which are named for their functions in females, but have similar processes, I guess you'd say, in males. So we have follicle stimulating hormone, which promotes egg development in females, and then sperm development in males. Luteinizing hormone is going to cause the testes and the ovaries to secrete their sex hormones. So for the ovaries to secrete estrogen and progesterone, and the testes to produce testosterone. We'll go into LH and FSH in much greater detail when we get to each reproductive system. But you should be familiar with hypothalamus, sends hormonal signals to the anterior pituitary. The anterior pituitary secretes hormones, LH and FSH, to the testes and the ovaries, which then they respond to the hormones to target cells. Okay. Again, we will go into that again next week. Now on to the major fun topic of reproduction today, meiosis. So meiosis is a unique kind of nuclear division that occurs only in the gonads, same process for males and females. It reduces the number of chromosomes in half. And so we end up with a haploid number of 23 chromosomes. We get 23 from the sperm, 23 from the egg, and then that will end up to 46, or 23 pair in the zygote. So some terminology, sister chromatids. So first we need to talk just a little bit about, we have mitosis and meiosis. So mitosis is what our cells do every day. So our skin cells are constantly undergoing mitotic division. So we lose a skin cell, we need to replace it. So mitosis is when we need to duplicate an identical cell to what we already have. And one of the things with mitosis, which we have many processes in place for, is to ensure 100% duplicity. So we don't want to vary our DNA once we're formed as an individual. We actually go to great lengths to make sure that DNA replication is identical. So skin cell A is a parent cell and it's going to make daughter cells. Those daughter cells need to look just like the parent cell. So mitosis is producing clones of cells. Meiosis is not, but before mitosis or meiosis is something called interphase. And interphase, like I said, is before mitosis or meiosis. And during interphase, we create duplicated chromosomes. So this is what I'll go with skin cell looks like right now. Before I make a new one, I need to duplicate identically the chromosomes that I already have. So this is the one I got from my dad, and this is the one I got from my mom, and then I duplicate those. When we get duplicated chromosomes, each of these, the blue one is the same as the blue one. These are called sister chromatids. If we're looking at a blue one versus a red one, those are non-sister chromatids. They're not the same. They'll contain the same genes for skin color or eye color, or hair color, or making muscle proteins. So they'll both code for the same purpose, but they aren't identical. Okay, so sister chromatids are identical DNA. Non-sister chromatids are the same genes, they're coding for the same function, but they're not the same. So in my case, I have one blonde and one black hair, okay? So those are the non-sister ones that they code for the same protein, but not the same, it's not identical. Homologous pairs of chromosomes, one member of each pair is from the father and one is from the mother. They look alike, but carry different alleles for the same genes. So non-sister chromatids within homologous pairs, chromatids of one chromosome are non-sister to other chromosomes in the pair. Non-sister chromatids exchange DNA during crossing over to increase genetic variability. So if we were in mitosis, so as an adult, we would duplicate these chromosomes. So we start off here, and then we duplicate during interphase. And then we would go through mitosis, and we would end up with two cells or two chromosomes that look just like the original set. In meiosis, the chromosomes get very close to each other, and they get so close to each other and they twist around each other that they swap sections of DNA. So now in meiosis, there is no chromatid that comes just from the maternal lineage or just the paternal lineage. They have swapped and made a new combination that does not exist in the parent. So diploid is the normal number of chromosomes in most body cells. So if you were to pull any cell from your body other than an egg or a sperm, they would be 46 chromosomes, 2 sets of 23. So 23 pair of homologous chromosomes for a total of 46. In a gamete, gametes are haploid. The number of chromosomes is halved, so there are only 23 chromosomes. And this is just another show of that. Okay. Sequence of events in meiosis. Meiosis consists of 2 nuclear divisions, meiosis 1, meiosis 2. DNA replication occurs before meiosis, so during interphase. The product is 4 haploid daughter cells, and each division involves 4 steps. So we have prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, and telophase 2. There are 3 figures that talk about meiosis. So this one is an overview of all of the entire meiosis, and then it goes through meiosis 1 and meiosis 2. For meiosis 1, so interphase is not a part of meiosis or mitosis, it is before, and this is where DNA replication occurs. So we had the original chromosomes and we replicate a second set. In prophase 1, this is where crossing over occurs. So we get parts of the maternal lineage and paternal lineage swapping places to create unique combinations. Then the second important event that contributes to genetic variability occurs in metaphase 1. We now have these pairs, they're not homologous anymore, they have new combinations. But the one that is primarily maternal versus the one that's primarily paternal, it's random as far as where they line up on the equator. Some will have mom on this side, maternal lineage, some will have paternal lineage. Then you go to the next pair of chromosomes and they're switched. And this forces an increase in genetic variability, and that when we pull those apart, some that are more like paternal lineage will go one way, and then some that are more like maternal lineage will go this way. So this way we don't end up with daughter cells that are mainly like mom, like maternal lineage, or grandmom, I guess, and more like grandpa, okay? We want a mix of as much mixing as we can get. So we get 2 daughter cells at the end, and then these are going to go on to meiosis 2, where now we do not replicate, because what would be the purpose? We want to get down to haploid. We do not cross over again, because then we'd just be undoing what we already did. So the genetic variability components are prophase 1, metaphase 1. Meiosis 2 is really just now getting to haploid. So it's starting with diploid. It's going to separate them along the equator, pull the chromatids apart, cytokinesis, split them in half. We end up with 4 haploid daughter cells. So meiosis 2 is really just about getting the genetic material down to haploid. Meiosis 1 is all about genetic variability. Meiosis 2 is how do we get to haploid. The next 2 slides are the same thing. They just have slightly better graphics, so I threw them in, and they have slightly better descriptions. Again, so interphase happens prior to meiosis. Prophase 1, crossing over occurs. So The chromatids get very close to each other and they swap some DNA. Metaphase 1, random assortment. So we've got blue on this side and then we have blue on this side. So random assortment along the equator also contributes to genetic variability. And here you can see the ones, so they have crossed over. So now none of these are like mom, okay? They're all brand new. We have crossed over, we have randomly assorted along the equator. Now anaphase is going to pull them apart so that we get, we actually now have a single set of 23 with twice as much DNA. And then these are going to be broken up into daughter cells. And then we go to prophase 2, metaphase 2, anaphase 2, telophase 2, and the whole thing here is genetic variability has been achieved in meiosis 1. So now in meiosis 2, it's to line them up, pull them apart, and create haploid cells. Meiosis compared with mitosis. Mitosis, we want clones. We want the exact same DNA. Okay, so the same DNA. These are going to be used for growth and repair. For meiosis, we need to create haploid cells. So reduce the number of chromosomes in half and introduce genetic diversity, which we do through prophase 1 in crossing over and metaphase 1 with random alignment. Okay, this is a table from your book that compares mitosis and meiosis. And then we are going to end with a quick little video on meiosis.
Humans produce gametes, eggs and sperm, through the process of meiosis. Here, we'll follow the production of male gametes by focusing on this cell as it goes through meiosis. Let's begin in the nucleus, where genetic information is stored in chromosomes. Most of a person's cells are diploid, with 2 sets of chromosomes. One set is from their mother, shown here in red, and the other set is from their father, shown in blue. Each maternal chromosome has a corresponding paternal chromosome. These matched pairs are called homologous chromosomes. During interphase, chromosomes are duplicated. Each chromosome now consists of 2 identical copies called sister chromatids. Zooming in, we see that each sister chromatid is made up of DNA wound around histone proteins. Each strand coils up into a tight helical fiber. As meiosis begins, a spindle forms and duplicated centrosomes start to migrate toward opposite poles of the cell. Back in the nucleus, the chromosomes are condensing. In meiosis, homologous chromosomes stick together in pairs. The close association of homologous chromosomes allows segments of non-sister chromatids to trade places. This recombination of maternal and paternal genetic material is a key feature of meiosis. After the spindle forms and the nuclear envelope breaks down, microtubules from opposite poles attach to each chromosome of the homologous pair, resulting in a tug-of-war. At metaphase 1, the chromosome pairs are positioned in the middle of the cell. The next stage begins when homologous chromosomes separate from each other and move toward opposite poles. Each chromosome still consists of 2 sister chromatids. This cell began meiosis with 46 chromosomes, but each daughter cell now has only 23 chromosomes. In meiosis 2, microtubules from opposite poles attach to the chromosomes, which then move to the center of the cell. Next, the sister chromatids separate, becoming full-fledged chromosomes that move to opposite poles. Nuclear envelopes reform, and each daughter cell divides into 2 cells. We started with a single diploid cell, and now that meiosis is complete, we have 4 haploid cells, cells with a single set of chromosomes. These haploid cells mature into gametes that can contribute to the next generation.
Okay. That is it for today.